Buenas tardes a todos. Muchas gracias por venir a la presentación del doctor Damien Godaux de la Universidad de Liège en Bélgica, que hoy nos va a hablar sobre su trabajo de su tesis doctoral acerca de la hidrogenasa y la regulación de la fotosíntesis en el alga Chlamydomonas reinhardtii. La presentación va a ser en inglés, así que espero que la disfruten mucho y le damos la bienvenida al doctor Damien Godaux. Thank you very much, Damien Godaux. It's a good pleasure to have you in this presentation for our students from the University of Mexico. And welcome to our university.
Thank you, Felix.
It is yours. Thank you.
Okay, thank you for the introduction, and I will thank you all and the organizers to give me the opportunity to give this talk. I will not try to share my screen, so if there is a problem, let me know if the presentation is clear. So do you have access to the entire screen now?
Yes.
Okay, good. So I will start. Yeah, so the title of the talk is this one, From Photosynthetic Regulation to Enzymo-Electrochemistry, 2 case studies to explore hydrogen bioproduction. So as mentioned in the title, our big topic today is the bioproduction of hydrogen. And the bio word is important as it indicates that hydrogen production is based on bioenergetic mechanisms inspired from nature. So it seems quite difficult to give a speech about hydrogen as a promising renewable energy carrier without starting with the decreasing availability of fossil fuels and the dramatic trend of climate change. Indeed, today, yeah, there is the laser here. All aspects of our society, from economic activities to daily entertainments, require a lot of energy. Fossil fuels are stores of chemical energy that are burned to produce power and heat and keep our society moving. This we know, but what does fuel look like? We have here the hydrocarbon molecule, which is a fuel. It is a long chain of carbon and hydrogen. And when this long chain come into contact with oxygen, and we give them a push, a huge amount of energy is released with some other byproducts, which are carbon dioxide and water. This is how our fuel works. And so far, such fuel is mainly fossil fuel like coal, natural gas, and oil. Our first problem is that these fossil fuels, by definition, are not renewable. And one day, maybe not so far, fossil fuels will running out. The second issue concerning fossil fuels is that the combustion leads to the release of large amounts of CO2 that, and it's difficult to deny, are known to induce changes in climate like global warming and wild and unpredictable weather conditions. There is then a general approval for the need of clean, renewable and sustainable technology to produce energy sources and solve the energy crisis. So the question is, can we make more fuel, but in a sustainable way? Hydrogen fuel is often pointed out as a promising candidate. When we refer to hydrogen, we actually refer to dihydrogen, the molecule made of 2 atoms of hydrogen, usually in a gaseous forms. Hydrogen is an energy carrier, which means that it is used to store energy produced from a primary process to move and deliver this energy that can then be converted later to another form. So this is the definition of an energy carrier. But why hydrogen is a promising renewable energy carrier. Firstly, because of its high energy content, which is the highest of any common fuel by weight, as we can see on this table. And for example, here is the value for hydrogen, and it is 3 times higher, for example, than the value content in diesel fuel. Secondly, hydrogen is really interesting because of its clean combustion into water. So indeed, once hydrogen has been produced, the energy present within the molecule can be released by reaction with oxygen, and we have only water as a byproduct. This reaction can be achieved by either traditional combustion engines or by devices called fuel cells. Another reason that makes hydrogen an interesting candidate is its wide range of possible applications, such as its use in decarbonized transport. For example, there is Toyota that is constructing and already selling hydrogen cars. There is a lot of buses in London that are working with hydrogen. And for example, in Germany or in Japan, there is high-speed trains working as well with hydrogen. Hydrogen can also be helpful to improve our energy integration with easy distribution of energy across different sectors or countries. It can also be used as a decarbonized feedstock for chemical process, for example, in metallurgy or in fertilizer industry. And finally, it is also known that batteries are not suitable in storing large amounts of electricity over time. A major advantage of hydrogen is that it can be produced from renewable and intermittent energy, such as solar panel or wind. And then this energy can be stored in hydrogen for extended periods of time. So hydrogen can therefore act as a buffer to improve our system resilience. Most of the hydrogen production strategy are currently realized by chemical extraction from fossil hydrocarbons, like we can see here in the red square, with the most important primary energy sources for hydrogen production being natural gas at 70%. So these methods obviously produce large amount of carbon dioxide. But hydrogen can also be renewable and decarbonized if producing using renewable and carbon-free electricity. For example, here, when the electricity comes from wind or solar panel in the process of water electrolysis. Today, only a small amount of hydrogen is generated from renewable energies, although that amount is set to increase in the future. So we can conclude that the search and development of new methods to produce renewable hydrogen are needed, and number of research on biohydrogen production have drastically increased in the last decades. Two biological processes have shown promising potential: the photochemical conversion of sunlight to hydrogen by a specific group of microalgae here, and the use of biological molecule to replace metal catalyst in electrolysis. So the two way of production here in green. Both these methods rely on a specific group of enzymes called hydrogenase. So the hydrogenase enzymes are metalloenzymes that catalyze the very simple and reversible reaction here. 2 proton plus 2 electron give the hydrogen molecule. As many living organisms use hydrogen as an energy carrier, Hydrogenases are widely spread among the microorganism diversity, with numerous representatives in the domain of bacteria, but also in some unicellular eukaryotic organisms. Despite their diversity in many respects, host, size, structure, electron mediator also, hydrogenases consist mainly in two phylogenetically distinct classes, the nickel-iron hydrogenase here with two atoms, one of nickel and one of iron in the active site center, and the iron hydrogenase with two iron atoms in the active site. They are generally presented as the result of a convergent evolution, and each class is characterized by a distinct functional core. But physiologically, it was shown that the hydrogenases are involved in various anaerobic bioenergetic processes. And the anaerobic word is important because one of the most important information here is that both the expression and the activity of the hydrogenase are sensitive to oxygen. In the first part of, I will not go further into details concerning the hydrogenase in general, but I will finish the introduction by giving a personal point of view, sorry. Of course, there is still a big gap to fill to obtain a commercially viable biohydrogen production system. Some major limitation needs to be understood and crossed, but this is the work of our work, the work of fundamental research to try to have a better, a deeper understanding of mechanisms that are involved to point out this limitation and give the information needed to allow the scaling up from laboratory to the industrial world. So in the first part of the presentation, we will discuss the interplay between photosynthesis and hydrogenase in the green microalgae Chlamydomonas reinhardtii. Almost 80 years ago, a group of researchers firstly reported the ability of some fermenting microalgae to evolve hydrogen upon a shift from dark anoxic condition to light. So this is quite strange because we have the hydrogenases that are sensitive to oxygen, so they are involved in the fermentation metabolism in anoxia. And when we switch on the light, there is a short burst of hydrogen that is produced. And it is possible because the enzyme is capable of interaction with the photosynthetic pathway, accepting electrons from water photolysis and resulting in the light-dependent generation of molecular hydrogen. It is now important to give a small introduction, a small recap about photosynthesis. So photosynthesis is the bioenergetic process by which higher plants and microalgae produce sugar. So they produce carbohydrates. Or do they do that? They combine carbon dioxide and water, and by the use of light energy, they produce this sugar that will serve as blocks to construct the cell or as fuel for the cell activities. So the photosynthesis is divided into two phases, the light-dependent reaction first. That's and the aim, the objective of this phase is to convert the light energy into chemical energy that are NADPH and ATP. So we have the photosynthetic pathway here with photosystem 2 that is able to use the light energy to do the water splitting of water, of course, and to split water into proton, oxygen, and electron. The electrons are injected into the photosynthetic pathway, and they go finally to the reduction and the production of NADPH. In this photosynthetic pathway, there is also the generation of a proton gradient here in the lumen, and this proton gradient is used to produce ATP. So in this phase, we have the use of light energy and water to produce oxygen and chemical energy. In the second phase of photosynthesis, which is the light-independent reaction, the chemical energy we produce in the first phase is used to fix the carbon dioxide into sugar by the Calvin cycle. And I will not go further into details for photosynthesis. We have enough information to continue the presentation now. In Chlamydomonas reinhardtii, which is a model microalgae to study photosynthesis by example, there is the presence of hydrogenase enzyme in the microalgae. So as I explained before, we have an hydrogen evolution in the dark due to the hydrogenase, which is a fermentative enzyme, but also in the light because of the interaction of the hydrogenase with the photosynthetic pathway here at the level of the ferredoxin. So just before the reduction of NADP to NADPH. The expression and the activity of the enzyme are sensitive to oxygen, and another details is that the hydrogenase need the expression of two maturation factors, high DF and high G, for the proper assembly of the active site, and then the activity of the enzyme. So our aim was to have a better understanding of the hydrogen metabolism in Chlamydomonas reinhardtii, because if we want to use this microalgae to turn the sunlight into hydrogen and to have a really nice way to produce renewable and sustainable hydrogen, we need to understand really well how all the mechanism work, how all enzymes are interfering into the production, and how the different photosynthetic pathway can interact with the hydrogen production. And also what we wanted to know if what is the role of such an oxygen sensitive enzyme here in the photosynthetic pathway, because the photosynthetic pathway, as we discussed, is a pathway that produces oxygen. So it's a bit weird that we have an oxygen-sensitive enzyme with an oxygen-producing pathway. So our first goal was to isolate new mutants impaired in the hydrogen metabolism. So of course, if we have new maybe we can have new proteins that are key proteins for the hydrogen production, and it is important to have a clear idea, a clear picture of how the system is working. So a first screening approach was set in 2004 by the group of Matthew Posevitz. And this screen was based on the ability of the algae to produce hydrogen upon the shift from dark anoxia to light. And when you put the algae colony in contact with a chromogenic sensor that is made, which is made of tungsten oxide, this sensor turns in blue in contact of hydrogen. We tried to do the same technique to isolate new mutants, but we could arrive to the conclusion that it is really time consuming. It is really invasive because we have to put the sensor directly on the agar plate with the microalgae, and it is also not really sensitive. But yeah, what is important to say is that in 2004, by the use of this chromogenic sensor, the authors could identify the mutant strain deficient for the maturation protein IDF. So we had an idea, which is to set up a new screen to isolate the mutant we wanted to find, and the new screen would be based on PS2 fluorescence. But what is the photosystem 2 fluorescence or the chlorophyll fluorescence? Fluorescence is a way that the chlorophyll have to release the excess of energy they can capture from the light. So when you have a lot of light that arrive on the chlorophyll, you have the photosystem 2 that is able to inject the electron into the photosynthetic pathway. But if there is not enough space to inject these electrons, the chlorophyll are able to release the excess of energy into fluorescence. So fluorescence is an indicator of the photosynthetic energy conversion, both in higher plants and in algae, because if the fluorescence is high, it means that the photosynthetic pathway is completely blocked and the photosystem 2 cannot inject the electron into the pathway. Then the fluorescence is high. And if the fluorescence decrease, it means that the pathway is no more full of electron, and that new electron can go through the pathway by the photosystem 2. So using the IDF mutant isolated previously in 2004, we could see that in a condition that are the condition where the hydrogen production is high. So in a shift from dark anoxic condition to light. So here at the time 0, our algae are in the dark and in anoxic condition. So the hydrogen is expressed. And we switch on the light here for 3 seconds. And what we see is that in absence of hydrogenase, the fluorescence is high. And in presence of hydrogenase in the wild type strain, we have a decrease of the level of fluorescence. And sorry, if we apply now saturating pulse, so we saturate the photosynthetic chain with a huge excess of energy, of light energy, we can see that there is no more space for photochemistry. So here in the mutant strain, the photochemistry is already full. The photosynthetic chain is already full. And here in the wild type, there is still some space for some electrons because we can have a higher value here. So this parameter is called the photochemical yield of the photosystem 2. And we can calculate it. And this is somehow the possibility of the photosystem 2 to inject the electron into the pathway. So we can see that we have a specific signature of the hydrogenase mutant compared to the wild type strain. Our question was, is this signature specific to anoxia? Is this an effect of the light intensity we used? And how this signature might be sensitive? We have been able to answer to all these questions because when we do the same fluorescence measurement but in oxic condition here, we can see that there is almost no differences between the wild type and the mutant strain. So this fluorescent signature here is specific to anoxia. This is not an effect of the light intensity because whatever the light intensity we used, we can see that the chain is always blocked. So this parameter here is almost close to 0, which is not the case for the wild type. So we can see that whatever the light intensity we use, there is no space to inject the electron into the pathway. And finally, we could see that this parameter, the photochemical tilt of photosystem 2, is directly related to the level of hydrogenase activity or hydrogenase expression. So with the calculation of this simple parameter here, we can have an access to the hydrogenase activity. And this is how we decide to publish our new fluorescence screening. But before to prove that the screening were efficient, we use as a proof of concept the finding of new mutants that are indeed impaired in the hydrogenase activity. So what we did is that we generate by random mutagenesis here a mutants library of about 2,000 strains. And we use our fluorescence screening to try to find new mutants. So we could directly on agar plate take a picture of fluorescence. Like this is what we have to do. And this picture gives us a value of the parameter photochemical yield of photosystem 2. And we find one strain that has a really low photochemical photosystem 2 activity. So because this photochemical activity is low, that means that the fluorescence is high. And the fluorescence is high because there is no hydrogenase. And indeed, we could find that the strain was a new strain, IG, that is deficient for the second maturation protein of hydrogenase. So the take-home message for this part is that we developed a fluorescence-based screening method to identify new players in hydrogenase activity and hydrogen metabolism. This new method is fast, non-invasive, and really sensitive. And we published this method in 2013 in the International Journal of Hydrogen Energy. Our second question, as I said, was to investigate the role of an oxygen-sensitive enzyme in photosynthesis. As we discussed, when we have the switch from dark anoxic condition to light, there is a short burst of hydrogen production in a wild-type strain. This wild-type produces hydrogen for a few minutes, maybe 2-3 minutes, before the hydrogen production stops. So, of course, this is far not enough to have a non-industrial production of hydrogen. And before our work, the main reason that was known for the cessation of this hydrogen production was the involvement of the oxygen produced by the photosystem 2 that will go to the hydrogenase and interrupt the hydrogenase activity and the hydrogen production. But we had another hypothesis that was our starting hypothesis. What if the hydrogen production stopped, not because of the effect of oxygen, but maybe because the electrons here from the ferredoxin stopped to go to the hydrogen through the hydrogenase, but decide somehow to go back to the NADPH to be involved in Calvin cycle and in the production of sugar. So, in other words, what if it was not an oxygen inhibition, but a competition between the hydrogenase and the Calvin cycle activities? It's really important because if we want to decide to produce more hydrogen with microalgae, we have to know which is the way that stops this hydrogen production. So to check if it was indeed a competition between hydrogenase and Calvin cycle activity, we had two tools: the photosystem 2 efficiency by the means of fluorescence, the same tools we use for the fluorescence screening, and the direct look at the level of hydrogen that is produced by the wild type. And for a wild type, we had these results. So we can see that indeed in 3 to 4 minutes, the hydrogen production here in blue drop to 0. But we can see that the photosystem 2 activity start to increase over the time. So again, the experimental condition are a switch from dark anoxic condition here in the dark where the hydrogenase is expressed to the light. So hydrogen evolution stops when the photosystem 2 efficiency increase. But our hypothesis was that this increase is due to the Calvin cycle activity. So what we use is the glycolaldehyde that is known as being a competitive inhibitor of Calvin cycle activities. And what we can say is that when we inhibit the glycolaldehyde, we have a continuous hydrogen production and without any stop for more than 10 minutes. So indeed, we can say that if we suppress the Calvin cycle, we continue to produce hydrogen. So in the wild type strain, there is a competition between the hydrogenase and the Calvin cycle activity. In another experiment, what we did is that we turned the lights off when the hydrogen production dropped to 0 in a wild type. We wait a bit, and here in black, we add glycolaldehyde and we turn on the light again. If the cessation of hydrogen production would have been due to the oxygen, we would have no more hydrogen produced. But if it was a competition between Calvin cycle and hydrogenase, by inhibiting the Calvin cycle, we would restart the hydrogen production. And this is the result we have. When we add the glycolaldehyde, we can start a new hydrogen production. So indeed, hydrogen production stops because of Calvin cycle reactivation prior to oxygen sensitivity. Okay, this is a really nice finding, and that was really new. But what we wanted to do is to go further to know why there is such competition between hydrogenase and the Calvin cycle, and how we could use this new finding to improve our capacity to produce hydrogen. Sorry. So here we have to go back to photosynthesis, and I have to give some more details. What I told you about the light-dependent reaction is that the light energy is converted into NADP and NADPH. This is true. And in this reaction, there is 1.3 ATP that is produced per NADPH. So the energetic ratio is 1/3. 1.3. But the energetic ratio that is needed for the Calvin cycle to fix CO2 into sugar is 1.5. So there is not enough ATP that is produced by the light-dependent reaction to be able to do the light-independent reaction and to produce sugar. So the photosynthesis needs alternative mechanism to produce an excess of ATP compared to the NADPH level. And these mechanisms are called the photosynthetic alternative electrons flow. In anoxic condition, there is few alternative pathways that are there to optimize the ATP versus the NADPH production. And one that is more famous is the use of NADPH in mitochondria with oxygen as final acceptor of electron. And in mitochondria, we use the NADPH to produce ATP. We have other pathways that go also through oxygen, and that will not allow the reduction of NADPH, but will continue to produce the proton gradient that will be used to produce ATP. And there is a last alternative pathway that is called the cyclic electron flow, which is a recycling of electron from ferredoxin, and they will be re-injected into the plastoquinone pool. So we have like a cyclic electron flow around the photosystem 1, and this cyclic electron flow will allow the generation of a proton gradient with and then ATP without a net production of NADPH. These are the photosynthetic alternative electron flow in oxic condition, but our condition was dark and anoxic, as I said. In this condition, because there is no more oxygen, there is no more mitochondrial activity, there is fermentation, and all these processes are known to produce a lot of NADPH without the production of ATP. So in this condition, there is a huge excess of NADPH, and there is very little amounts of ATP within the cell. So the Calvin cycle is, of course, completely blocked in dark and anoxic condition. And our suggestion is that when we switch on the light, the electron don't go to the Calvin cycle because it is completely blocked, and the hydrogenase produce the hydrogen to allow the generation of proton gradient and then ATP, and to restore the ATP and NADPH balance. So to prove that the key parameter for the reactivation of Calvin cycle was indeed the generation of proton gradient and then the ATP production, we use CCCP, which is an uncoupler of the proton gradient generation. Destroying this proton gradient, we could not synthesize any ATP. And what we observe is that without ATP, we have, as here again, a continuous production of hydrogen. So what we could say is that we, in a wild type, we go to the hydrogenase pathway as long as Calvin cycle is not reactivated. We can see it with the glycolylate inhibitor. And when we destroy the ATP generation, we also do not go, the electron, they don't go to the Calvin cycle, and they continue to go to hydrogenase, which is a proof that this is the competition with Calvin cycle, and that the Calvin cycle reactivation is ATP dependent. As I said, in anoxic condition, we have two pathways that are still there, because all the pathways to oxygen are not possible. So we have the hydrogen production and the cyclic electron flow around the photosystem 1. And this electron flow is PGRL1 dependent. So PGRL1 is a protein that allows the re-injection of electron from ferredoxin to the plastoquinone pool. So we had different set of mutants from for hydrogenase activity, the Ig2 mutant that we isolate in the first part of the talk with the fluorescent screening, and also a mutant, a PGRL1 mutant that is not able to have this cyclic electron flow. And what we can see is that in both mutant strains, simple mutant strains, we have a delay in the possibility to reactivate the Calvin cycle, but we still have a Calvin cycle activity. But when we cross these two strains to have a double mutant, the PGRL1 Ig2 deficient strain, we lost this ability, this capacity to reactivate the Calvin cycle. So our conclusion is that Calvin cycle reactivation is prevented in absence of both hydrogenase activity and PGRL1 cyclic electron flow. What we did as a funny experiment was to mix the double mutant with a wild type strain in a condition where the photosynthetic reactivation was often needed. So in a 3-hour dark, 3-hour light cycle, So we mix both these strain, the wild type and double mutant, and we check every day the amount of each strain in the mixing culture. And what we see in that, in absence of both PGRL1 and IG2 photosynthetic way, the mutant cannot reactivate the Calvin cycle, and then by the time it disappears from the culture, which is a proof that Chlamydomonas keep both this pathway, because it is important in condition where photosynthesis reactivation is needed. So as a take-home message, we demonstrate that hydrogen production stops because of Calvin cycle reactivation prior to oxygen sensitivity, which is really important if we want to use this system to produce hydrogen, and that either hydrogenase activity and PGRL1 cyclic electron flow is required for Calvin cycle reactivation. So this is the end of the first part of my talk. These data have been published mainly in plant physiology, but also in 2 other papers. Here, one in collaboration, with Dr. Bart Geisel and Dr. Fabrice Franck, and I would like to thank them a lot for this collaboration, and also all the collaboration we did after. And another paper with Sophie Clovet and Fabrice Rapaport that we published in the Journal of Biological Chemistry, and which also was a really cool collaboration. These fundamental studies provided very important information on the understanding of the hydrogen metabolism in microalgae. This information that might be very useful to greatly improve the hydrogen photoproduction. This is in fact become somehow a reality, as in 2018, this paper published by the group of Iftach Jacobi, and that is complementary to ours, because they have the same finding that hydrogenase activity is outcompeted by carbon fixation before inactivation by oxygen. So this is the result we demonstrate in 2015. But what they did more is that using short light and dark cycles, the author can maintain the hydrogen production, so here in green, and they can maintain this hydrogen production for more than 50 minutes. So it's much more than if we go back to the result I present, it's here, sorry. Here we have something like 3 or 4 minutes before it's go to 0. And because of this new finding, they could extend this hydrogen production up to 50 minutes. Last year, the same group described the hydrogen production of the HS14 mutant. This mutant possesses an hydrogenase enzyme that is fused, that is merged with another enzyme, the superoxide dismutase. Superoxide dismutase is an enzyme known from another fundamental studies to improve greatly the hydrogen production. So this HS14 mutant, as we can see, is able to produce hydrogen for more than 5 days without stopping, which is quite really impressive, because at the beginning we were at 3 minutes, and now with the use of all the fundamental information we obtained, we can go to more than 5 days. And a last paper, which has been published in November 2020, so it's really new, where the group of Sergei Kosurov use a cycle of 1 second light and 9 seconds dark, and you doing this 200 times, we can see that they can have a continuous hydrogen production. So what they are doing is that they are switching off the lights so often that you cannot produce enough ATP to reactivate the Kelvin cycle. And in this condition, you can increase a lot the hydrogen production. Yes, okay. This is the end of the first part with the hydrogen production from photosynthesis and from microalgae. In the second part of the presentation, we will discuss the ability of hydrogenase enzyme to replace metal catalyst in K devices for the use of hydrogen in our economic system. K devices such as electrolyzer and fuel cell. Electrolysis currently accounts here. Electrolysis currently accounts for around 5% of the global hydrogen production. This hydrogen occurs in electrochemical device called electrolyzer here. And the electrolyzer use an electric current at the anode to split water into its constituent parts, which are oxygen, proton, and electrons. Protons and electrons go to the cathodic part of the electrolyzer, where they are combined to produce hydrogen. So if renewable or carbon-free electricity is used, for example, with wind or solar panels, the hydrogen has a zero carbon footprint and is known as green hydrogen. Fuel cell is another electrochemical device that in fact do the opposite reaction. It consumes hydrogen here at the anode to extract electron and proton. The proton, so it creates electricity and it uses in fact the energy that is stored in hydrogen to produce electricity. Proton and electron are rerouting to the cathode and combine. This electron here will go to the anode and will react with oxygen to produce hydroxide anion. Hydroxide anion will react with the proton that are produced at the anode to form water. So here we use hydrogen and we produce electricity and only water in fuel cell. But the major issue using electrolyzer and fuel cell is the use of metal catalysts such as platinum or iridium for the electrochemical reaction. These metal catalysts are not renewable as they are present in a defined quantity on Earth. They are located in specific regions so that their exploitation might be subject to geopolitical problems. And finally, their extractions provoke pollution and social issues. There is therefore huge interest to find a solution to replace such metal catalysts. So the question is, could enzyme, and here could hydrogenase replace such metal catalyst in electrochemical device? So indeed, hydrogenase could solve most of the problem I just present. Indeed, hydrogenase will be purified from microorganisms that might be cultivated anywhere, solving the renewable and the location issue at the same time. The results I am about to show you in this part demonstrate the use of hydrogenase in such condition, but they are not directly, unfortunately, data obtained in our lab. I cannot present our specific work due to confidentiality clause, but the papers I will present give a very good overview of the work which is done with hydrogenase enzyme in electrochemistry. To describe the following result easily, I will start this section by a small explanation about electrochemistry and especially about one specific and useful technique of electrochemistry called cyclic voltammetry. Cyclic voltammetry is a technique that you will change the potential of the electrode, and you will have a look at the current that appear. And when you look at the anode, you will have what is called an anodic oxidation with a positive current, because you are going to create some electrons. And when you look at the cathode, you have a reduction with a negative, with a negative current here, because you are using electrons. So it's a reduction. The first paper and the first question, is could hydrogenase replace the metal catalyst in electrolyzer? So here in electrolyzer, you're gonna use the hydrogenase on the cathode and to combine electron with proton to produce the hydrogen at the cathode. And this is the work that has been done by the group of the Professor Jan Sappel in Germany. They used the nickel iron hydrogenase of cyanobacteria called Synecocystis. They purified the hydrogenase and they put the enzyme on an electrode, on a cathode. And what you see using the cyclic voltammetry is that at a certain potential, you have a current, a negative current that appears. Negative current, that means you have a reduction, the reduction of proton by the use of electron into hydrogen. And they try to do this reduction at different pH. And you can see that the more the pH is acid, the better the hydrogen production is, because of course with acid pH you have more protons, so you have more substrate to produce the hydrogen. So here what we see is that we can have an hydrogen production at a cathode by the use of an hydrogenase. We can have the same question with fuel cell. Could hydrogenase replace the metal catalyst in fuel cell? And of course, the answer is yes. So here is a work of the group of Antonio de Lace from the University in Madrid, which we collaborate with, the lab. So they published in 2005 the structure of the enzyme, and they have seen that there is a dipole, an electric dipole, because of the structure of the enzyme, and they use this dipole to have a really strong link, a covalent bond between the hydrogenase and the carbon electrodes. So here you can say in a few years later, in 2007, they published a paper where they coated hydrogenase on carbon nanotube, and they have an efficient hydrogen oxidation. So indeed, because here in fuel cell, hydrogen is used at the anode, we can see that we have an anodic current oxidation with a positive current, which means that we are using the electrons that are in the hydrogen. And we can see here where when the hydrogenase is coated on carbon nanotube, that we have a really stable current within more than 1 month. So this electrode is really stable, and you can use the enzyme in a fuel cell. We can even go further and have a look if the enzyme could replace both catalysts, for example, in a fuel cell, because we have seen that the hydrogenase could oxidize the hydrogen to create electricity, but we can also replace the metal catalyst we have at the cathode by another enzyme, the bilirubin oxidase, to have a completely enzymatic cell. And this is what did the group of Elisabeth Lejoux in Marseille. where they create a hydrogen-oxygen enzymatic fuel cell. So here is just a picture on microscopy where you can see that this is the carbon nanotube before you coat the enzyme on, and here is the enzyme that are present in the electrodes. Here is the hydrogenase enzyme. And so having the cyclic voltammetry technique, we can see that we have a bioanode that is working with the hydrogenase that is creating a current. And at the biocathode, we have the same result with bilirubin oxidase that is functional, that is using the current produced at the bioanodes to reduce oxygen into water. So it's working really well. And here, what we are seeing is the dark curve, and we see that they lost only 30% of the activity within 17 hours of activity of both enzymes. So they could replace both catalysts with both metal catalysts with enzyme. So yeah, here is just some numbers to say that we can see that depending of the temperature, of course, but we have a high number of milliampere that are produced in this condition. And what I wanted to say is that it's going really close to the power that are needed to have an industrial use of enzyme into fuel cell. So this paper is really close to fill the gap between the laboratory and the industrial world. And this is the last paper I am going to present. Is that also what we can do is that we could replace the anode in an electrolyzer with the photosystem II. So as I explained at the beginning of the introduction, the photosystem II is able to do the water splitting. So it uses light to split water into proton, electron, and oxygen. And this is the work that has been done by the group of Erwin Reisner in Oxford, and they linked the photosystem II to nitrogenase to do the water splitting and to produce hydrogen. So this is exactly what has been done. So you have photosystem II anodes, and when you put light on it, you have the water splitting. You create a current, and this current can go to the cathode, and the hydrogenase use these electron and proton to produce hydrogen. So here, what you do is you store the light energy you use here into hydrogen. So just with enzyme, you can go from light energy to hydrogen as energy carrier. Okay, yeah, so it's again the same result where you can see that both photosystem II and hydrogenase are working independently and also together. So now is the end of my talk, and I will finish this presentation with two anecdotes. The first one is Jules Verne that referred to hydrogen already back in 1874. The French writer expressed something that a long time after his death might become reality. He said, "I believe that water will one day be employed as fuel, that hydrogen and oxygen, which constitute it, used singly or together, will furnish an inexhaustible source of heat and light, of an intensity of which cold is not capable." So hopefully he will be right. Another and last fun fact is that hydrogen has the atomic number of 1, and its standard atomic weight is 1.008. So the World Hydrogen Day has been derived from this fact and is on the 8th of October of each year, which is funny actually because it's my birthday. So maybe I am not working on hydrogen by chance. Yeah. So I would like to thank all my collaborators during the last 10 years in the lab, of course, of Professor Claire Remacle and Dr. Pierre Cardel. Also in the lab of Dr. Fabrice Franck and especially Bart Geisels, and as well Dr. Philippe Lorch, which is full of really, really interesting idea about hydrogen. I thank you for your attention. Okay, Felix.
Thank you very much, Damien. It was very nice. So maybe somebody from the people that is listening can want to ask something. If anybody has any question, you can write it either in the chat or you can ask directly to Damien. I think we have enough time to put some questions.
I am checking the chat. I don't know if I have access. Okay. No question?
No, maybe nobody has any question.
So clear.
No, well, me, I have a question. It's about the rate of let's say the productivity when it is produced in the algae, in Chlamydomonas. Does it have any role, this hydrogen molecule in the alga?
No, it's just a way to evacuate the excess of energy. So to avoid the over reduction of the photosynthetic pathway. And it's the same at the beginning. The hydrogenase is an enzyme that is working in the fermentative pathway. And in fermentation, what is really important is to oxidize the reducing equivalent to maintain the fermentation and to maintain the ATP production. And somehow it's the same in the photosynthesis, and it's more or less all the time the activity of the hydrogenase is to avoid the over reduction of the pathway by evacuating this excess of electron and to maintain an ATP synthesis. So yeah, the hydrogen go directly out of the cell and never come back. And this is what is funny, is that at the beginning people were somehow talking, okay, this is really nice that Chlamydomonas and other microalgae are producing such hydrogen, and they were not even, or somehow not even wondering, okay, but why is this enzyme here? So they just say, okay, we have an enzyme sensitive to oxygen, so it stops because of oxygen, which at the end was not true. And the people were not even wondering why is this enzyme there.
And it disappeared from the green line of plants, right? There's any plant with this hydrogenase?
Nope, there is not. No. So we guess that because Chlamydomonas and other microalgae that contain hydrogenase are microalgae that might frequently encounter anoxia in the natural environment, which is not the case of higher plants. So they keep the hydrogenase both for the fermentative pathway, but also to be able to reactivate the Calvin cycle activity when the light comes back.
There are a couple of questions in the chat. One is somebody is asking, what about a sexual cycle in such conditions of growth? Chlamydomonas.
I don't understand the question.
I don't understand the question either.
Neither do
because,
I.
Yeah, so in such condition of growth, is it the anoxic condition or is it the normal condition? Because what is important to understand is that this hydrogenase activity is really like a transition from one anoxic dark condition to the light, but it's not related to the sexual cycle of the microalgae.
Okay, so it's not maintained for long period. No? So
yep, exactly.
So there is another question, and it's how long will it take to have this industrial production as a current process?
It is the question, because when I started the master thesis 10 years ago on hydrogen production in Chlamydomonas, we were already introducing the paper with this aim of the grail that might be the hydrogen production, which might be bring to an industrial level, and we are so far from it. So I don't know how many time it will take. What I know is that it's worth it to have funding to continue to investigate this way of producing renewable energy, to continue to have this option, because if we stop now, we will not go further. So we have to continue. But I cannot say if one day we will have a breakthrough with someone finding something really interesting, really important. I hope so.
Okay.
Yeah. And thanks for the so clear and very interesting comment. I hope that you like it, even if my English is not good enough for such a long talk. But yeah, I hope you all understood.
Creo que no hay más preguntas, Félix.
Okay.
I don't know if there's another questions. No sé si los estudiantes tengan alguna otra pregunta, la pueden escribir en español y bueno, intentaremos traducirla para Damien.
Okay. I will try to understand Spanish.
Okay.
Hello. If there is some more time, I would like to ask a question as well.
Please.
Yes, Damien was explaining that the hydrogenase is an oxygen-sensitive enzyme, and we have seen through his results that actually this sensitivity to oxygen is kind of lost in physiological condition, so into the cell of Chlamydomonas. I was wondering if he knew the reason why the hydrogenase into the cell is actually not it's not sensitive to the oxygen evolution.
Thanks, Fabrizio, for the really good question. To my opinion, is that when you have the photosynthetic pathway and the photosystem 2 that is feeding the chain with so much electrons, you have the active site of the hydrogenase enzyme that is full of hydrogen, proton, and electron that are created at a really high speed. So I should have started the answer by saying that the oxygen inhibits the enzyme by attacking the active sites. So my idea is that if the active site is working and that it's producing hydrogen all the time, every second, every milliseconds, maybe the oxygen cannot enter within the active site. So I think this is because the enzyme is working that the hydrogen cannot attack the active site. But if you purify, for example, the enzyme, and that in such condition the enzyme is not working, then the hydrogen can attack the active site and inhibit the enzyme.
Por ahí hay otra pregunta, Félix, en el chat. ¿Tienes apagado tu micrófono, Félix?
If Damien, can you activate your camera to see you?
Sorry, I didn't know it was.
And there are a couple of questions more. There is somebody asking if can hydrogen production be an economic way of producing energy? So maybe in terms of the money that is needed to produce hydrogen like this.
With enzyme, not yet. But with the fuel cell and the electrolyzer are already a thing. I mean that there is a lot of companies all around the world that are producing hydrogen at a huge level by the use of electrolyzer, for example, and it's just few percent of the total hydrogen amounts that is produced, but it's already a reality. With the enzyme, we still have to improve our yields, to improve the current density, the enzyme production, because of course with enzyme it's really small amounts each time, for each purification. We have a lot of work to do, but what I would say is that there is it's something that I think really interesting. It's more a political idea, but my feeling is that there is no there is a problem with the way hydrogen fossil fuel are used in our society compared to the renewable energy, because we are so used to work and to function with this fuel that we don't even let a chance, I mean a normal chance. So what I would say is that, okay, we go to the zero level and we see for every fuel what is the benefits and what are the costs. For example, with the diesel, we have costs for the health of people, and we put the costs of health that the diesel produce on the society into the cost, and then we can say, okay, diesel is this cost and hydrogen is this one. So we can have a real comparison of all the energy on the market, and maybe we would with this have a better idea of how far we are with the renewable energy, and for example with hydrogen.
Okay, sorry, there are other questions, a couple of questions more, but there's people thanking you, saying thank you to you because it was an excellent talk, and also if there is any relation between the production of hydrogen, I mean, and the production of lipid droplets in this algae, and also if there is any similarity between the metabolism of this. Maybe the question is related to hydrogen production and this energy metabolism with Euglena's metabolism.
Sorry, you lost me for 1 second, and I could not understand the question. Can you repeat?
If the metabolism of Chlamydomonas is similar to the one of Euglena in that sense, or if there is any relation between lipid droplets and the production of hydrogen.
This is a nice question, and I will not, unfortunately, be able to answer. What I know is that we have people in the lab working and trying to produce hydrogen with Euglena. Maybe she will be able to give an answer to this, but I cannot pronounce myself because I'm not sure about what to answer to this. So I cannot compare Euglena and Chlamydomonas.
Other questions? This enzyme hydrogenase, what's the lifetime in the electrocell?
So what I present with the work that have been done at the University of Madrid in the group of Antonio de Lace is that they could maintain the hydrogenase activity for more than 1 month, which is really impressive. This is one case with one precise hydrogenase, because of course there is a lot of different hydrogenases and some are better than others, and it's also depends of what you want to do. But for example, using the photosystem 2 on electrode, the life is so much shorter. It's something like from 10 to half an hour. So it's really a short period of life.
And there is another question maybe related to this, is how easy is to isolate or to produce the complex of photosystem 2 for these cells, for these electrocells?
Yeah. Looking at the literature, in the bibliography, there is a different protocol to produce and to isolate the photosystem. And also it might depend of what you want to do, because you can isolate the cure, just the few main protein of the photosystem 2. And you have a pure cure of photosystem 2, but maybe this unit will have a better activity, but for a shorter period of time. And another option is that you do I would say a larger purification with a lot of contaminant, with piece of the chloroplast and that kind of stuff. And you add a mediator, which might do the link between the photosystem 2 and the electrodes. And then you keep the photosystem 2 in its environment, and you allow to have a much higher period of time alive. Yeah.
And there are other questions. For example, there are many questions, Damien. But one question more is, could you explain more how they relate ETR to the activity of hydrogenase, and if it was done in anoxia?
So we related the electron transfer rate to hydrogenase and
the activity of hydrogenase. The ETR, the electron transfer rate, and the activity of hydrogenase. How are they related? How do you?
What we did is that with the fluorescence measurement, you can have the photochemical yield of photosystem 2. You can multiply this parameter with the light intensity you use, and you have a relative electron transfer rate. So this is a formula that is well known in the photosystem measurement. and having access to the size of the antenna of each photosystem, the photosystem 1, the photosystem 2, you can have a correction and really have the good measurement of how many electrons are involved into the photosynthetic, are injected into the photosynthetic pathway. This is for the how we measure the photosystem 2 activity. And for the hydrogen production, we have a probe that directly measure the concentration of diluted hydrogen, so in micromolar. It's not directly in micromolar. It's a probe that is based on electrochemistry, and which, according to the level of hydrogen that is dilute, give us a level of current. So the more you have current, the more you have hydrogen, and you can do the correlation between the level you have and the amount of hydrogen, so you know at which speed the number of nanomole are produced per minute, and depending of how much cells you put into your measurement cell, so you can have a direct access of how much hydrogen molecule are produced. And of course, there is 2 electrons by hydrogen molecule, so you know how much electrons are directed to the hydrogenase pathway.
There is another question more. Does the light intensity really matter when applied on the isolated photosystem 2 in the electrode system? So which light intensity is best there?
Yeah, it's a really good question, and it's really important. It's not only the light intensity, but also the quality of the light. So we know that using a red light, which excites specifically the photosystem 2, we have a better current, but also using a pulsing light, for a reason we are still not sure, we have a more extended period of activity. So with the same photosystem, using different lights, different way to apply the light, we have different results. So it's an entire part of experimentation to do just with this parameter.
Nice. And maybe just to finalize, there is another question. What are the study perspectives in hydrogen production, and what's next in the research on the production of hydrogen with
I would say I am working on the hydrogen production with the electrochemical device with electrolyzer, trying to replace platinum with the hydrogenase. I would confess I miss a lot the photosynthetic regulation studies that I did at the beginning during my PhD thesis. But I would say that if we want to go closer to the industrial production of hydrogenase, this is our best options, to my opinion. And for example, the work that I presented that has been done in the group of Elisabeth Lojou in Marseille, with the combination of the hydrogenase and the bilirubin oxidase, they try to identify all the parameters that are really important. So the temperature, the substrate diffusion limitation, the product inhibition, the structure of the electrodes, the link between the hydrogenase and the electrode, and using and studying all these parameters, they could go really close to the level, the power that's are required for an industrial production. So maybe we are not that far to cross a line and to maybe soon being able to compete or to be really close to the hydrogen production level that are required.
Great, that sounds exciting.
It's a lot of questions, a lot of things. Hay otra pregunta ahí, Félix.
Perdón.
Maybe the last one.
Okay. Yes, I'm sorry, I did not see it. There is a last question. What are the main advantages of hydrogenase deficient mutants according to the fluorescence kinetics?
I really don't understand.
Maybe the question is more related to
the study.
Yes.
So the mutants, the mutant without hydrogenase activity don't have any advantages because in normal laboratory oxic condition there is no difference, and in anoxic condition the strain is less able to reactivate the photosynthetic pathway. But the way we are working is that we create some mutants, we delete some pathway, and we say, okay, without this, we have a decrease. So maybe if we improve this, we have a better, and this is how it's working all the time, at least for now. We are saying, okay, we have this mutation and we have this impact. We have this mutation, we have this impact. What if we combine? Okay, we have these results. So there is no really advantages of creating the hydrogenase deficient strain. But this is a way to say, okay, this pathway is involved. So maybe we can play on this pathway to change the hydrogen production level.
Okay, so the development of this technique or this strategy to analyze the hydrogen production was really like the important step, no, to study the hydrogen production.
Yeah. Okay.
Well, I don't know if somebody else has any other comment, any professor like Dr. Hugo, do you have any comment before?
No, I don't. No, sorry, Damien, I'm going to speak in Spanish. Félix, no sé si ya se puso el link, no estoy viendo al mismo tiempo el enlace, perdón, para subir las constancias, o si alguien quisiera una constancia, ya no lo vi. Ahí está el enlace, Félix, para los de Facebook, ¿verdad? Digo, los de Zoom. Sí. ¿Es ese enlace? Sí. Y en YouTube ya se subió.
Sí, también.
Sí. Ok, correcto. Thank you. No, it was a very nice talk, Damien.
Thank you. I hope you enjoy it.
I mean, yeah, it's very nice, very interesting. Yeah, thank you very much.
Nice.
I don't know.
It was really a pleasure to give this talk.
Okay. No sé si hay alguna otra pregunta por los estudiantes o los que están viéndonos por YouTube. I don't know if you have another questions to Damien. I don't know if I say your name correctly.
Yeah.
It's okay?
Yeah.
Thank you. Yeah. No sé qué más, Félix. I don't know. No sé qué más, Félix.
Well, para los asistentes de la universidad, el link es para que puedan obtener una constancia por la asistencia, y el contacto del doctor Damián está en la presentación. Si pueden acceder a ella en la versión de YouTube, pueden comunicarse con él en caso de que tengan alguna duda acerca de su tema, quieran contactarlo por alguna cuestión. Y bueno, creo que este es el final de la presentación. Yo creo que a nombre de toda la universidad y toda la comunidad estudiantil podemos darle las gracias al doctor por la presentación. Thank you very much, Damian. It was a very nice presentation. I think all students are benefiting from this. It was very nice to listen to you. Thank you.
Thank you, Felix. Thank you.
A todos los estudiantes, no sé si los que están y que pudieran y quisieran abrir su cámara para saludar al doctor y darle las gracias. Damien, we are going to open our video so we can say thank you very much. Thank you, Damien. This is our students and our professors from the university. Thank you very much. And
thank you.
Thank you very much from Mexico.
Thank you.
Damien. Where are you right now, Damien? In
Liège, still in Liège with Felix at the university.
All right.
Yeah.
Thank you very much.
Thank you.
Gracias, muchas gracias. No sé qué más, Félix, y
Bueno,
eso sería todo por el día de hoy.
La presentación. Muchas gracias a todos. Hasta luego.
Gracias,
Hasta luego.
chicos.